Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, we were learning about cellular signaling, in which we have covered various signaling pathways which are activated due to the activation of GPCRs or the receptor tyrosine kinases. So, we are done with those two particular receptor proteins. Today, we are going to discuss about some other receptor proteins such as the cytokine receptors or the receptors for certain hormones which signal through enzyme coupling, but they do not have intrinsic enzymatic domain. So I hope you remember when we were discussing about cellular signaling. When we started discussing this cellular signaling topic, we discussed that for a cell to be able to communicate with other cell or for a cell to receive certain signal, it needs a receptor protein on its cell surface, right? So the receptor proteins that are present on cell, depending upon which type of mechanism they used, so that they can switch on and off certain gene transcription, depending upon those mechanisms that are used by them, there are three different categories of the cell surface receptors. The first ones are GPCRs, second one are the RTKs, and third one are the inotropic receptors, right? So these three categories we discussed in detail. The inotropic receptors have been discussed in neurobiology series, while the first two, GPCRs and RTKs, that we discussed here in the cell biology series. So we discussed about how in the case of GPCRs, the receptor protein they use G proteins, the GTP binding proteins, as a mechanism to switch on and off the transcription of gene. So we discussed about the intracellular signaling cascades, which uses molecular switches so that they can switch on and off certain gene transcription. So molecular switches are the proteins which can get activated or inactivated with the help of some extracellular signal. So according the cellular need, to the requirement of the cell, those molecular switches will get switched on or they can get switched off so that they can switch on and off certain gene transcription, right? So this that is why they are known as molecular switches. So the way we have our switch box, the control button, so that we can switch it on, so that we can switch on the light bulb or the fan. In the same way, those molecular switches they can be activated so that they can start the transcription of certain gene or they can switch off the transcription of certain gene. So we discussed about the two major categories of the molecular switches, the GTP binding protein and the kinase phosphate modules. Okay, kinases and phosphatases modules. So we discussed about GTP binding protein. So if those GTP binding proteins are used as the molecular switches for primarily to act after the receptor is activated, such receptors are known as G protein coupled receptors because they are being coupled with G protein. Means the activation of that receptor protein is going to activate the G protein, which is a molecular switch, right? Or, on the other hand, the activation of the receptor protein due to the extracellular signal, this activation can result into the activation of certain inactive enzyme. And the inactive enzyme can downstream do some phosphorylation jobs to activate certain protein, or it could be coupled with G protein again. So we discussed, so let me just write it down so that it will be clear to you all. So we discussed that receptor proteins are crucial so that the extracellular signal is being received, right? So those receptor proteins, these are the transmembrane proteins which will have extracellular domain. Okay, so they are going to have extracellular domain. They are going to have a transmembrane domain which is going to hold the whole protein in the plasma membrane, and they are going to have intracellular domain. Okay, so the extracellular domain, the job of the extracellular domain is to receive signal. It is going to receive the extracellular signal. It is going to interact with ligand, and that is going to promote some conformational changes. And those conformational changes will be mirrored with the help of transmembrane domain towards the intracellular side, towards the intracellular domain of this receptor protein. So transmembrane domain not just holds the protein in the plasma membrane, but it also has its job in mirroring the conformational changes that are being done in the extracellular domain towards the intracellular part. So the intracellular domain is the one which is going to result into the downstream signaling. Okay, so the activation of this intracellular domain due to the conformational change that has occurred from in the extracellular domain and is being mirrored by the transmembrane domain, the intracellular domain will now do some downstream signaling job. How? Because it is getting activated. So once it is activated, so depending upon to which this intracellular domain of receptor protein is being coupled, to which type of protein it is being coupled, it is divided into three categories, right? So those three categories, all of them, they need or they uses different types of mechanisms, different types of mechanisms, so that the downstream signaling would be carried out. So this intracellular domain could be coupled with G protein. That is why such receptor molecules are referred as G protein coupled receptors. Okay, so this intracellular domain could either have intrinsic receptor tyrosine kinase activity, okay, or it could be coupled with some enzymes. So that is why these are known as enzyme coupled receptors. Such receptor proteins are known as enzyme coupled receptors, out of which the major family, the major group is the receptor tyrosine kinases. There are some other enzyme coupled receptors as well, but this is the major group, and that is why we discussed that in the cell biology, in the cellular signaling series, right? And the third type of receptor proteins where the intracellular domain could be coupled with ion channels, they are known as ionotropic receptors. Okay, so all of those receptor proteins, they use different type of mechanisms. So what does GPCRs use? They use activation of G proteins as the mechanism to do the intracellular signaling part. The receptor tyrosine kinases use the transphosphorylation, means the tyrosine kinase activity, so that the downstream kinase cascade could be switched on. So if in the enzyme coupled receptor, either the downstream kinase cascade could be switched on, okay, so that the intracellular signaling goes on, means the kinase cascade molecular switch is being switched on, or they can also use the help of some G proteins. They can also, receptor tyrosine kinases, they can also switch on the G proteins downstream, which are going to do the intracellular signaling job. Okay, for example, we learned in RAS MAP kinase pathway, the activated receptor tyrosine kinase will activate RAS protein, which is a monomeric GTPase. So it is also a G protein, and it is going to activate the some downstream phenomena. It is going to do some downstream phenomena, which is going to result into activation of certain genes, right? So either of the molecular switches could be used with the help of RTK. G protein coupled receptors simply use G proteins as the molecular switch, while RTKs or the enzyme coupled receptors, they can use either of the molecular switches. But the ionotropic receptors, they are very different because they are going to use ion channels. They are directly, so activation of those receptor protein will directly open an ion channel. So what does this ion channel do? So ion channel is going to exchange some ions. So this exchange of ions will, it will result into unequal distribution of the ions across the plasma membrane, and this unequal distribution of ions will change the membrane potential of the plasma membrane. Okay, and the change in membrane potential will result into the change in the electrical properties of the membrane, and that is going to result into some downstream signaling. So ionotropic receptor uses the change in membrane potential due to the opening of ion channel as a mechanism to ensure that downstream signaling occurs. So these three receptor proteins are very different, and we have discussed about GPCRs, we have discussed about ionotropic receptors, we have also discussed about RTKs, the RTKs which can activate kinase cascades such as MAP kinase modules through G proteins such as RAS proteins. So we have discussed both of them. Okay, so the direct kinase cascade activation will be seen in PI3 kinase signaling, right? Or the kinase, even the G protein are also using the G protein are also switching on kinase in case of RAS MAP kinase module. Okay, so activated RTK will activate RAS protein, which is a G protein, which will activate MAP kinase module. okay, MAP kinase module, which is again is going to activate some kinase cascade. So either of them could be activated with the use of RTKs. But the receptor tyrosine kinases that we discussed till now, they have intrinsic kinase domain, but there are some receptors which also come under the category of enzyme coupled receptors that we are going to discuss from today. Okay, those are the receptors that are being coupled with the cytoplasmic tyrosine kinases. So we have in the enzyme coupled receptors, we have learned about RTKs, which is the largest group of the enzyme coupled receptors. And now we are going to discuss about some receptors, which are the receptors for cytokines or hormones, which are going to be associated with the cytoplasmic tyrosine kinases. Okay. So the major differences, so in the enzyme coupled receptors, there are two categories. The first one we discussed receptor tyrosine kinases, and second we are going to discuss about the receptors which are being coupled with the cytoplasmic tyrosine kinases. Okay, so both the as the even so receptor tyrosine kinases, the receptor intracellular domain itself has the tyrosine kinase activity, but in the case of cytoplasmic tyrosine kinases, the intracellular part is just there, it is going to get activated, and this intracellular part, it is being coupled with an enzyme, with the enzyme which is inactive when the signal is not there. So one this will be activated when signal comes and binds to this receptor protein. So the job of intracellular domain in this case, in the cytoplasmic tyrosine kinase associated receptors, in this case the intracellular part only does the job of being attached or being associated with inactive cytoplasmic tyrosine kinase. Okay, so it has only one job. Whatever conformational changes are being done, that are being conveyed on the intracellular side as well, and it is being associated with some tyrosine kinases. So so basic difference is that RTKs have intrinsic tyrosine kinase domain. Okay, so the tyrosine kinase activity is itself present in the receptor, but in the case of in the case of cytoplasmic tyrosine kinase associated receptors, the other cytoplasmic tyrosine kinase is being coupled with the intracellular part. So this is the only difference between both of the receptors. And that is why, but as both of them have tyrosine kinases being coupled with enzyme, so whenever extracellular signal arrives in both of the cases, whatever downstream activities are going to be done, that resembles a lot. So the way the tyrosine kinase is going to be autophosphorylated, and it provides docking site for SH2 domain containing proteins. In the same way, when the cytoplasmic tyrosine kinase get activated, it also phosphorylates the intracellular domain of the receptor protein, which is going to create phosphotyrosine residues for the SH2 domain containing protein. So whatever downstream process, whatever downstream cascade is going to be seen, that will resemble a lot in those cases, because the enzyme that is going to do the activity is the tyrosine kinase. So both are the tyrosine kinases, means they are going to add the phosphate group over the tyrosine residues. So whatever mechanism the cell has to follow, it is quite similar. Okay, that is, and that's why you won't be confused by those categories. So the inactive cytoplasmic tyrosine kinases, which can be coupled with those receptors, they could belong to SRC family. They could also belong to integrin family. Okay, and they also belong to some other families. There are some other kinases such as JAK, Janus kinases, and some of them that we'll be discussing in some other lectures. So the point of discussion for today is the JAK-STAT signaling. So this is also a cytoplasmic tyrosine kinase which is being coupled with a receptor. The receptor which is which is receiving cytokines, as well as it can also receive some certain hormones. So today we are going to discuss about the JAK-STAT pathway, Janus kinase signaling pathway. Okay, so let me write it down for you. Today we are going to discuss JAK-STAT pathway, JAK-STAT signaling. Okay, so till now we have discussed about GPCRs and RTKs, the receptor proteins which are cell surface receptors. In the same way, the receptors for cytokines, they are also cell surface receptor, but that does not come in any category of the GPCRs and the RTKs. So the GPCRs, they usually do the signaling for vision, signaling for smell, right? The rhodopsin receptors. So those are the GPCRs. The RTKs, we learned that they are very important and they are going to activate some kinase activity downstream, and they are going to result into cell growth, cell survival, right? So in the case of immune responses, when the mediator called cytokines, which is a chemical mediator which is being released by the activators immune response, that cytokines, they are also, so the receptor for those cytokines are present on the cells, and that is why those receptors are being named as cytokine receptors. So they simply have the name cytokine receptors. So those cytokine receptors, okay, let me just write it down sideways. This cytokine receptor, this is not just a receptor for cytokines, but it is also a receptor for certain growth hormones or even the hormones like prolactin. Okay, so this does not only work for cytokines, but it can also receive growth hormones, okay, or hormones like prolactin. Okay, so prolactin is a milk stimulating hormone. So whenever prolactin arrives and it attaches to the receptors present on the breast cell, it is going to result into some downstream activity, which is going to result into formation of milk proteins. Okay, so the prolactin, this hormone, and certain growth hormones and cytokines, they can go attach themselves to the receptors, and such receptors, they are being coupled with the intracellular with an intracellular enzyme, with a cytoplasmic enzyme known as Janus kinase. Okay, so JAK is an enzyme known as Janus kinase. So this is also a tyrosine kinase, which is going to phosphorylate tyrosine residues, which are present on the intracellular domain of this receptor protein itself. Okay, so just like any other receptor tyrosine kinases, those cytokine receptors, they are also present in the dimerized form. So this is the cytokine receptor. So this cytokine receptor, which is present in the dimerized form, it will come and it will bind to the receptor protein, and it is going to result into the dimerization of receptor. Okay, so the pathway that is being followed, I already told you the pathway is quite similar. So on the plasma membrane, two receptor proteins, okay, which are being coupled with JAK, they will come closer, they will get dimerized, okay, due to the cytokine being received by them. So cytokine, when it will dimerize the receptors, and those receptors, they are being coupled with the enzyme JAK. Okay, so this JAK, the physical property of those kinase domains which are present in this protein JAK, it will result into the activation of this JAK. So the activated JAK, it is going to now phosphorylate itself. So what will happen, that this JAK will phosphorylate the kinase domain of other. So this happens, this is just like the transphosphorylation which happens in the RTKs, and this kinase domain will phosphorylate this the tyrosine residues present on this JAK. So let me just write it down for you. So let's assume that this is JAK1 and this is JAK2. So JAK1 will phosphorylate tyrosine domains on JAK2, and JAK2 will phosphorylate tyrosine domains on JAK1. So this will create, okay, so this will create the activated domains due to physical proximity. This will create phosphate residues, okay, phosphotyrosine residues over the Janus kinase. So this will fully activate the Janus kinase domain, which is present here, and the full activation of Janus kinase will result into the phosphorylation of this intracellular domain. Okay, this will result into phosphorylation of the tyrosine residues which are present on the intracellular domain. So what will happen? This JAK1 will phosphorylate tyrosine residues on the intracellular domain, and JAK2 will also phosphorylate tyrosine residues present on the intracellular domain. So now here you can see that phosphotyrosine residues are being formed on the intracellular part of the receptor, of the cytokine receptor. So now this phosphotyrosine residue which are being formed, due to the JAK, the Janus kinase, to those phosphotyrosine residue, now the SH2 domain containing proteins can bind. So which proteins are SH2 domain containing proteins? The proteins which has SH2 domain and which are going to get bind to this JAK associated receptors are known as STAT. Okay, the signal transducer means this is the protein which is going to perform the transduction of signal, means the relay of signal downstream, and it is the activator of transcription. So the name itself makes quite clear the function of the script. Okay, so the name itself, the stat name itself, signal transducer and activator of transcription itself makes clear what kind of function stat is going to do. So it is going to relay the signal downstream, means it is signal transducer, and it also does the activation of certain genes, and it is also referred as activator of transcription. So stat proteins, they have SH2 domains, and that is why stat proteins they will now bind to this phosphotyrosine residues which are present on the intracellular part of the cytokine receptor. So this stat, there are at least six different types of stats present in mammalian system. So either both of them could be similar, okay, both of them could belong to one family, or they could belong to some other family. Okay, so either this could be stat 1 or stat 2, means it can belong to same category. So there are six different categories, minimum, at least six different categories present in the mammals. So both of them could belong to same category, or they could belong to the different category, both of the stat molecules. Okay. So when the stat molecules, they attach to this phosphotyrosine residues present on the intracellular part of the receptor. So now what will happen? The Janus kinase, which is a kinase, it is going to phosphorylate those stat residues as well. Okay, so this is the next step, the phosphorylation of the stat residue. So phosphorylation of tyrosine residues present on the stat. So now stat will also get phosphorylated. So phosphate residues will be added over the stat, so the phosphorylation of the stat residue will release the stat. Okay, so it will release the stat from the intracellular domain, and they will be released in the cytoplasm. Okay, so now as they are released in cytoplasm, what will happen? The SH2 domain which was bound to this phosphotyrosine residue, okay, that is now free. It was bound to phosphotyrosine residue present on the receptor. Now this phosphotyrosine residue is free. So what will happen? So let's assume that this is the phosphate group of the phosphotyrosine residue. Sorry, my bad. Let's say, so what I was trying to explain is that due to the phosphorylation of stat by the JAK kinase, so JAK kinase is phosphorylating both of the stat molecules. So the phosphorylation event of the stat will release the stat in the cytoplasm. Okay, so this will result into the free SH2 domain. Okay, so now the STAT protein, which is now in the cytoplasm, it is phosphorylated, and the SH2 domain, which was previously bound to the intracellular part, intracellular phosphotyrosine residue, that is now free. So what will happen? This SH2 domain will bind to the phosphotyrosine residue of the other STAT protein. So what will happen? So this particular STAT protein, okay, this is STAT1 and this is STAT2. Okay, so both of them they are phosphorylated, and that is why they are being released in the cytoplasm, right? So both of them are phosphorylated. Okay, and both of them have a free SH2 domain. Let's assume that this is the SH2 domain. So SH2 domain, I'm going to denote it with a different color, violet color. So let's assume that both of these are the SH2 domains, which was resulting into binding of the STAT to the phosphotyrosine residues of the receptor. So now those SH2 domain, they are free. So what will happen? This SH2 domain of STAT1 will bind to the phosphotyrosine residues of STAT2, and the SH2 domain of STAT2 will bind to the phosphotyrosine residue of STAT1. So that is why the STAT1 and STAT2, both of the molecules, they will form a dimer. Okay, so they will form a dimer. Here I'm trying to draw the dimer. Okay, so this dimer, which is being formed by STAT1 and STAT2, will be due to the both of them having the SH2 domain. Okay, so this SH2 domain will be bind to the phosphotyrosine residue of STAT1, and let's say that this SH2 domain of the STAT1 will be bound to the phosphotyrosine residue of the STAT2. So this will create a dimer, which could be either a homodimer, okay, if both of the STAT molecules belong to the same category, or it could be heterodimer if the both of the STAT molecules belong to different categories. So it could be homodimer or heterodimer depending upon the category or type of the STAT molecules that are being adhered to each other. So this STAT, which is being now released in the cytoplasm and it is formed dimer, so this STAT is referred as the activated STAT. So this phosphorylated STAT is the activated STAT, and now it will translocate to nucleus. So it will translocate to the nucleus, and this translocation of activated STAT to the nucleus will result into the binding of the STAT molecule to the cytokine responsive regulatory sequence, which is present inside the DNA of the cell. And the binding of STAT to its designated place will result into the activation of transcription of certain gene. And this is how the JAK-STAT pathway is going to work inside the cell, and it is going to activate the responses of a cell, which are meant to see which are meant to be seen after, or to be observed after the cytokine release. So whenever cytokines are released and they attach to the receptors, those receptors, by which they are going to work, these receptors are associated with JAK proteins, the Janus kinase, and they are going to take the help of JAK-STAT signaling, so that the transcription of certain genes, the cytokine response genes, gets activated. Okay, so I have a schematic for it, again taken from Bruce Alberts. So this nice schematic will explain you everything. But before that, let's have a look at a table which is again given in Bruce Alberts, where you can see that how different extracellular signal protein, they act through the JAK-STAT signaling. So all of those signal proteins, they are going to work through JAK-STAT signaling. Means the receptor proteins for those signals, such as interferon gamma, interferon alpha, or the prolactin, as I told you, and some growth hormones. The receptors for all of them, they are associated with Janus kinases, and they are going to take help of JAK-STAT pathway to give some cellular response. Okay, so for example, the prolactin, which is a milk stimulatory protein, the milk stimulatory hormone. So this prolactin will it will be so the receptor for those prolactin, they will be associated with JAK or JAK2. So there are four types of Janus kinases. I forget to mention that: the JAK1, JAK2, JAK3, and TYK2. So these are the four different types of Janus kinases that can be seen in the biological systems. So these four Janus kinases and six different types of stats exist in the at least six different types of stat exist in the mammalian cells. Okay. So, so this is how the JAK stat signaling is going to result into some cellular responses. So the prolactin will be received by the prolactin receptors. So the prolactin is being received by prolactin receptors, and those prolactin receptors, they can be associated with either Janus kinase 1 or Janus kinase 2, and they are going to result into the activation of stat known as stat 5, that is going to result into the activation of the genes which are going to do the production of milk proteins. Okay, and that is why prolactin is known as milk stimulatory hormone. Okay, so that in the same way I explained about prolactin, the other signaling molecules also, these are the other signaling molecules which work through JAK pathway. So interferon gamma, the receptor for interferon gamma could be associated with JAK1 or JAK2, and they activate STAT1, which is going to activate macrophages. The interferon alpha, okay, again both of them, they are released by the activated immune system. So the receptors for interferon alpha, they are associated with TYK2 and JAK2. Those Janus kinases are being activated, and they will activate STAT1 and STAT2, which will increase the cellular resistance to the viral infection. The erythropoietin, this hormone, it will be the receptor for this will be associated with Janus kinase 2, JAK2, and it is going to activate STAT5, which is going to result into the production stimulation. It will stimulate the production of erythrocytes. So this erythropoietin, it is very crucial for the production of erythrocytes, means the RBCs. Okay, so I already explained prolactin. Prolactin receptors will be associated with JAK1 and JAK2. They will activate STAT5, and that is going to stimulate milk production. The growth hormones, the receptors for the growth hormone receptors will be associated with JAK2. They are going to activate STAT1 and STAT5, which is going to stimulate growth by inducing the IGF-1 production. So what is IGF-1? IGF-1 is insulin-like growth factor. Okay, 1. So I already explained how the insulin-like growth factors, they work through RTK activation. So this is how the growth hormone will work through JAK-STAT signaling, which will produce IGF-1, which is going to work through RTK signaling. Okay, fascinating, right? So the GM-CSF, which is also known as granulocyte macrophage colony stimulating factor. So this GM-CSF is very crucial, and I'll discuss this about when we start immunology lectures. So the receptors for CSF will be associated with JAK2, and they will activate STAT5, and this GM-CSF, it is important for production of granulocytes and macrophages, and that is why it is quite evident from the name that it is a granulocyte macrophage colony stimulating factor. Okay, so these are the different signal proteins which will activate different JAK and STATs, and that they are going to work through JAK-STAT signaling. Okay, so let's summarize the whole lecture in this schematic itself. So here you can see the schematic explains the cytokine signaling, which is mediated. So cytokine mediated signaling, which uses JAK-STAT pathway. Okay, so this is how. So the cytokine receptors, they are present on the plasma membrane of the cell, and they are associated with the Janus kinases, the JAK proteins. Okay, so those kinases, they are being simply associated, stably associated with the intracellular part of the receptor. So whenever cytokine or any growth factor will come, so it will result into dimerization of the receptor protein, which will bring the intracellular domains of the receptor proteins together, and that will bring the kinase domains of the JAK proteins together. So this physical proximity of kinase domain will activate the kinase domain, and it will result in the phosphorylation of the domains of the other tyrosine residue which are present on the other kinase. So, for example, this JAK1 will phosphorylate the tyrosine residues which are present on the other protein, other JAK protein, and the kinase domain of this JAK protein is going to phosphorylate the tyrosine residues present on the first JAK protein. So now the phosphotyrosine residues of on the both JAK proteins are created. So this phosphorylation of tyrosine kinases will result into the full activation of kinase domain, and that is why now this kinase domain is going to phosphorylate the intracellular domain, the intracellular tyrosine residues present on the receptor. So this receptor is a protein, right? So it has the intracellular domain and it will have some tyrosine residue. So on those tyrosine amino acids, those kinases, the Janus kinases, so they have the tyrosine kinase domain which is now fully activated. So they are going to phosphorylate those tyrosine residues. So once those tyrosine residues are phosphorylated, so to this the SH2 domain containing proteins called STAT, the signal transducer and activator of transcription proteins will attach because they have the SH2 domain. So those SH2 domain will bind to the phosphotyrosine residue, and now those STATs will be phosphorylated with the help of Janus kinases. So the phosphorylation of Janus kinases, so the phosphorylation of STATs with the help of Janus kinases will result into the release of those STAT proteins inside the cytosol. Now they will be released because the phosphorylation will do some conformational change, will weaken this interaction of SH2 domain with this phosphotyrosine residues on the receptor proteins, and instead those SH2 domain of those STAT proteins will adhere to the phosphotyrosine residue of the other STAT protein. So this will create a dimer of STAT proteins, which could be a homodimer or heterodimer. In this case, this is a homodimer. So now this activated STAT, this activated phosphorylated STAT, this dimer, it will translocate itself in the nucleus. Okay, so and in the nucleus it will form a transcription regulatory complex with some other protein, with some activator proteins which are going to bind to the cytokine responsive cis regulatory sequence which is present in our DNA. So this sequence, you can see easily, you can gaze easily from the name that it gets transcribed when the cytokines arrive at the site. So that is why they are named as cytokine responsive regulatory sequences. So now they are going to result into the transcription of the target genes which are crucial for the cell survival in the case in this particular case, right? So this is how the cytokines they take the help of JAK-STAT pathway to transcribe certain genes. So we are done with JAK-STAT pathway. So we have learned about GPCRs, RTKs, and JAK-STAT signaling, which uses the cytoplasmic tyrosine kinase residue. So that is why the signaling pathway, it is very similar to the receptor tyrosine kinases. The only difference is that the cytoplasmic tyrosine kinases JAK, they are associating themselves to the in the cell, the DNA which is going to transcribe, so the gene which is going to transcribe this cytoplasmic tyrosine kinases, it is different. So the peptides are different, and the receptor peptide and the tyrosine kinase peptides are different, and this is not the case in the receptor tyrosine kinases. In the receptor tyrosine kinases, this intracellular part will itself has a tyrosine kinase activity. So that is the only difference. Rest of the pathway is quite similar, and this is JAK-STAT pathway because it uses a kinase protein named as Janus kinase and the signal transducer protein known as STAT. So we are done with JAK-STAT signaling. In next class, we'll learn about TGF-beta signaling, and we have to yet learn about a lot of pathways such as Wnt signaling, Notch delta signaling. So till then, see you. Bye bye.